So microsporogenesis, as the name indicates, so what happens? Production of microspores from pollen mother cell or microspore mother cell, or where these two are present, as we have discussed in the last session, sporogenous tissue which is present in the sporangium. So maybe you have remembered the sporangium structure with the multiple layers, yes, where they contain the sporogenous tissue like this. Okay, tissue which is made up of pollen mother cells or microspore mother cells. So what happens? Just consider a single pollen mother cell. So the single pollen mother cell, if we consider this as a single pollen mother cell, so I'll write PMC or MMC. You should write in a abbreviated, you should not write in a abbreviated form. Please expand it and write pollen mother cell and microspore mother cell. So one more thing, they are haploid in nature. Okay, they are 2N. They undergo meiosis. We know there are two stages in meiosis, meiosis 1 and meiosis 2. When a single cell undergo, what happens? Meiosis 1, they form two cells. They again undergo meiosis 2 and they form 4 cells. So beginning, the parental cell is 2N. When they undergo first meiosis, yes, second meiosis, N, but the result is 4 cells, right? So remember the meiosis division what we have studied in our first year. Okay, so what is the result of meiosis? One cell, if they undergo meiosis, they form 4 cells and which are haploid in nature. So here also the same thing happens. When a pollen mother cell or microspore mother cell which is diploid in nature undergo meiosis, what happens? They give rise to microspore tetrad. Okay, so as we know, tetrad means 4, and why it is called microspore tetrad? So because they have attached together. Okay, they have not separated, they have compacted, they are placed in a compact thing, so hence it is called microspore tetrad. So later what happens? Due to the dehydration in the anther, so these microspores, or none other than, it is also called as a pollen tetrad also, later it is called as pollen grains, they form separately or they separate together. Okay, so these are now called as our pollen grains. Okay, so what happens? From a single pollen mother cell, there will be formation of 4 pollen grains. Before formation of pollen grains, there is a structure called microspore tetrad, or it is also called as sometimes pollen tetrad also. Why? Because of there are 4 pollen grains or microspore, but what happens? They are not separated. Okay, they are placed in such a manner compactly, so hence it is called as microspore or pollen tetrad. After the anther, when they get dehydrated, okay, so what happens? This pollen tetrads within the pollen grains, or within the pollen tetrad, pollen grains are present. No, they separate and form like this. So what is the fate of a one pollen mother cell? 4 pollen grains. So this production of pollen grains, okay, or microspores from polar mother cells or microspore mother cell is called as microsporogenesis. And all these our pollen grains are haploid in nature. This is very important, okay, during fertilization process. So we should understand what is the ploidy of each cell, which is very important. This is what microsporogenesis, okay. So next, what happens, there should be development of this pollen grains, okay, for fertilization. So this single celled pollen grain is not efficient one, okay. So to fertilize the egg which is present in the gynoecium, our pollen grain should be get matured, okay. So that is the next step now, maturation of pollen grain. Okay, so before going to the maturation of pollen grain, we'll study the structure of pollen grain. Pollen grain, or important thing, this pollen grain is also called as male gametophyte. Remember, okay. Yes, we'll draw the structure now. The pollen grain structure, especially they are made up of two walls. Okay, so they are made up of two walls. So the decorative hard outer wall, we call it as this wall. We call it as exine, which is outer wall, okay. And smooth inner wall, we call it as intine, inner wall. And there are some gaps between. In some places this wall is not continued. Which wall? Exine wall is not continued, because see, can you see here, the exine wall is discontinuous in some places. That discontinued place we call as germ pore, okay. And we know usually what they contain, a single nucleus, because as we have studied during the microsporogenesis, the cell contains single nucleus, okay. The pollen grain contains single nucleus, and this is the cytoplasm of the pollen grain, okay. So what pollen grain? It is not yet matured. It is not yet matured, only the simple structure of pollen grain, okay. Early stage of pollen grain consists of what? Mainly two layers, outer exine, inner intine. This exine wall is made up of some substances or chemicals called as sporopollenin, which is very hard in nature and which doesn't reacts with any acids or enzymes or organic materials which is present outside the environment. Hence makes sporopollenin the harder wall, okay, the harder compound, and that is the composition of exine. Why? Because of sometimes the pollen grains are exposed to the environment. There will be rain, there will be drought, there will be sunlight exposure, okay. So when they exposed to such environmental conditions, so they may like get destroyed easily, okay. And we know this is the only one male gamete which is produced in the plants, which helps to like continue their progeny after the fertilization process. So to maintain their viability or liveliness, so what happens, this exine wall is made up of a hard withstanding chemical, that is sporopollenin, okay. In the same way, this intine Okay, intine also made up of a chemical or substances called as pectin and cellulose, okay, which is made up of pectin and cellulose and make them smooth. Okay, and how like there will be formation of a pollen tube, as we said, the formation of a pollen tube. How there will be formation of pollen tube? So like this, a tube is formed, extended. This extension will be helped by the germ pore. Okay, the function of germ pore is the formation of pollen tube. During or at this gaps only, what happens at the spaces here, which is discontinued space of exine, from where this pollen tube will be emerged, where they can reach the ovary and undergo fertilization process. So they help gamete to get transferred in the female reproductive part of a flower. Okay, this is what the structure of a simple non-unmatured pollen grain. Okay, now this pollen grain is get matured to perform fertilization now. Okay. We'll go to the next process, that is development of pollen grain, or it is also called as maturation of pollen grain. As we have studied the structure of pollen grain, what it consists: single cell, okay, single cell, and this single celled pollen grain undergo not repeated, we can write only mitotic division. Okay, and they divide into two cells. Okay, they divide into two cells, that is irregular nucleus, one, and another is spindle shaped nucleus. So this two cells, which is after the mitotic division, we call it as vegetative cell, or it is also called as tube cell, and this is generative cell, okay, which consists of generative nucleus, and here it is vegetative or tube nucleus, and other things are similar. Exine, intine remain same. So the one thing what differs here, the single celled is now converted into two celled stage of a pollen grain. What are those two cells here? The bigger one called as vegetative cell or tube cell, which contains their respective nucleus, and the smaller we call it as a generative cell, and which contains its nucleus respectively. Okay, what is the fate of vegetative cells? Vegetative cells helps in the formation of pollen tube. Okay, generative cells, what happens? So sometimes this two-celled stage is converted into three-celled also. And how? Because this is our large tube nucleus, tube cell with or vegetative cell with vegetative nucleus, and this is our small generative cell, which mitotically this nucleus divides and forms two male nuclei, or sometimes it is also called as male gamete. Okay, yes, So hence it is called as generative. What happens? This two male nuclei now will help in the fertilization process. They go and fuse with the egg to perform the fertilization. So what happens sometimes in angiosperms, maximum, that is 60% of plants in the anthers, the pollen grains will dehisce or will come out, disperse out at the two-celled stage. Sometimes remaining 40% of angiosperms where the pollen grains are dispersed at the three-celled stage. Now you understood? So now this two pollen grains, which is two-celled or sometimes a three-celled, now it is called as matured pollen grain, okay, which is ready to perform the fertilization now. Okay, so what happens? By the meiosis, pollen grains are formed from the pollen mother cell. Here, what happens? A single-celled pollen grains, they undergo mitotic division. They divides into two cells, that is vegetative, bigger vegetative and smaller generative cells. That is two-celled one. Later, what happens? That two-celled converted into three-celled. How? The generative cell, they divide into two male nucleus, or in turn it is also called as male gamete. So what happens? The upper vegetative cell or tube cell helps to form a pollen tube, and those pollen tube which carries these two male nuclei for the fertilization process. Okay, 60% of angiosperms, they disperse the pollen grains at the two-celled stage. Remaining 40%, they disperse the pollen grains at the three-celled stage. Okay, so this is what the development or the maturation or microsporogenesis. So now about the pollen viability we are going to study. Okay, so what happens? Any pollen grain, after its dehiscence, their function is to go and reside on the stigma of the gynoecium for the fertilization process. Okay, when they disperse from the anther, they go and reside on the surface, okay, of stigma. Okay, due to some temperature or humidity, so what happens? There will be sometimes an immediate germination of pollen grain, or sometimes may not be, based on the external things. Okay, so what happens? So viability means from the time of disperse or the pollination and its germination. Germination means pollen tube carrying those two male nucleus. This is what called as a pollen germination, which reaches the ovule and get fused and they perform fertilization. Okay, so this time duration is called as viability. Just imagine this pollen grains viability, or they are inactive for 5 days. They stayed on the surface of the stigma for 5 days. After 5th day, that is 6th day, they start germinated. Okay, they are active. So this 6 days or 5 days we can consider it as a viability of the pollen grain. Viability is nothing but the capacity to germinate. Okay, capacity to form the pollen tube which contains the male gamete for the fusion. This we call it as viability. Understood? So this may be sometimes a months also, sometimes some hours, sometimes years together. Like our cereals, pulses like wheat, barleys and all, they have their pollen grains have a viability period of 30 minutes. Sometimes some leguminaceae family plants, they have a viability period of 1 month. Because of the viable nature, those pollen grains, scientists store those pollen grains in liquid nitrogen at 190, minus 190 degrees Celsius. They store, and the storage unit they have called as pollen bank. How there are seed banks where the seeds are used for future like production of plantation of plants. So here also, because of their viable nature, so they stored the pollen grains of some species with the help of liquid nitrogen at minus 196 degrees Celsius. So they some institutions they produced the pollen banks. Okay, so this is the viability, and as advantages and disadvantages, many knows, like the major advantages of pollen grains are they are very nutritive. Uses we can say, because of nutritive nature, okay, they prepare sometimes pollen tablets also, or sometimes pollen syrups. Okay, yes, which is given to sometimes humans also, like some athletes or race horses, because they need energy to perform well. They supplement them with the tablets or syrups which is made by the pollen grain, because of the nutritive nature. Because as I told you, the pollen grain consists of one big cell and a small cell. The big cell we call it as vegetative cell. Vegetative cell consists of stored material, food material. Okay, that food material consists of nutrients which is used to make tablets and syrups also. Like disadvantages are they cause allergies. This is not uses. Uses is only nutritive in nature. This is disadvantages. Disadvantages are they cause allergies, like some chronic respiratory disorders. And one example is asthma. Okay, infection of like lungs. Within the lungs there will be infection which is may be caused due to the allergic condition. Some more persons, maximum persons are like allergic to the pollen grains. Okay, pollen dust, they are very allergic, which cause them asthma or a chronic respiratory disorder. So these are some uses as well as disadvantages, as well as pollen viability. Based on pollen viability, this year in our practicals we are going to study practically, we are going to understand the pollen viability by performing the pollen germination experiment. Okay, so practically you will get to know what is actually the pollen viability. Okay.